Thank you. First, I'd like to thank the Welch Foundation and Kayvon for that, for inviting us, for me to give to come here and talk to all of you. It's really exciting opportunity. We don't typically get a room full of chemists to talk to, so we're really excited about that. So thank you for that. I'm also really excited to share with you some of our work in pancreatic cancer. As you may know, pancreatic cancer is the deadliest human cancer and currently the second leading cause of cancer death in the United States. So today I'll be telling you about our efforts to reverse translate some observations from rare survivors of pancreatic cancer to potentially some solutions. So we are squarely in the era of precision immuno-oncology because we have in the past several decades made fundamental advances in solving a really critical question for immuno-oncology, namely, how does the immune system recognize cancer and intrinsically self-derived tissue? We now know that mutations cause normal cells to become cancerous, but as cancer cells divide and grow, a fraction of these mutations also produce new proteins on the cell surface called neoantigens, and these neoantigens serve as novel proteins that can be recognized by T cells, allowing T cells to recognize cancer cells as foreign and kill them. We also know that the ability of these T cells to recognize these neoantigens, these T cells can really unleash powerful immune responses in patients with clinical impact, thus unlocking a suite of possibilities to expand these cells in patients and change clinical outcomes, either directly in vivo by expanding these cells with antibodies or vaccines, or expanding these cells ex vivo through cell therapies and then reinfusing them back into patients. And essentially launching the next era of immune therapies, we believe precision immune therapies that, unlike the last generation of oncology drugs, which have been largely static drugs, this next generation really offers the possibility to treat cancer with dynamic, intelligent, and evolving medicines. Now, a fundamental challenge for this vision has been that the current class of immune therapies are effective in only about 20% of human cancers, leading the field to question whether the remaining 80% of cancers are immunologically invisible, thus rendering an immune therapy just not possible at all. And one of the cancers that is sort of the prototypical immunologically invisible cancer has been pancreatic ductal adenocarcinoma, or PDAC. Now, PDAC has been considered to be immunologically invisible because it has very few mutations and thus fewer mutated neoantigens. And because of this, very few infiltrating T cells in tumors, rendering these cancers largely insensitive to the first generation of immune therapies that Emily mentioned, the immune checkpoint inhibitors, and broadly typifying the other 80% of human cancers that, like PDAC, are immunologically cold and insensitive to immune therapies. Now, as I mentioned, PDAC is also a cancer with very high unmet medical need, the second deadliest cancer in the United States in 2025, so more deaths from PDAC from many of the common cancers that you might hear about: breast cancer, colon cancer, ovarian cancer, prostate cancer, second only to lung cancer. And the survival rate with current treatments, which include surgery, chemotherapy, and radiation, remains only about 10%, so really underscoring the need for novel therapeutics. Now, this observation that PDAC is immunologically invisible really stemmed from this fact that the immune response rate to immune checkpoint inhibitors was largely zero in PDAC. Thus, perhaps PDACs are immunologically invisible. But for the past several years, we've been challenging this notion, saying, well, is this really true? And by studying these rare survivors of PDAC, these 10% of patients that, unlike other PDAC patients, survive long term. So really asking the question, how do they do it, and how could we potentially replicate this long term? And what we have learned now by studying these patients for the past 10 years or so is that these long-term survivors of PDAC, we believe, mount natural T-cell immunity to PDAC with primary tumors enriched in many CD8 T-cells, that although PDACs have very few neoantigens, in fact recognize somatic passenger mutated neoantigens that you can identify a priori based upon their unique structural features. This immunity is amplified by other cells in the microenvironment through the formation of lymph node-like structures within tumors. And excitingly, you can in fact deliver these neoantigens to other patients with RNA to induce many T cells in these patients and phenocopy this survivor state. And in fact, this phenomenon of these vaccine-induced T cells, they in fact have quite superior longevity and function, so ideal characteristics for a cancer vaccine. So by studying these rare survivors of PDAC in the last 10 years, we've now successfully led the clinical discovery and development of 2 new potential immunotherapies for pancreas cancer, the first PDAC RNA vaccine, as well as a first-in-class recombinant cytokine. And today, I'm excited to tell you about our work on the first pancreas cancer RNA vaccine. So this really started about 10 years ago, really with this one question, well, is there natural immunity to PDAC? And to answer this question, what we did was we assembled 2 cohorts of PDAC patients, a cohort of short-term survivors of PDAC with a median survival of less than 1 year after surgery, and a stage and treatment-matched cohort of long-term survivors of PDAC with a median survival of 6 years after surgery. And we asked the question, well, what was really different about these 2 cohorts? And what we found was that this exceptional survival of these rare patients could not be explained by unique treatment, pathology, or somatic genetics. But one of the most striking differences that we had seen in these patients was they had primary tumors that were enriched in about 12 times greater densities of activated CD8 T cells, which are the killer T cells that protect our body against viruses as well as cancers. And this is shown here by this representative immunofluorescence image where all of these yellow cells are CD8 T cells in a long term compared to a tumor of a short-term survivor. And you can just see by eye that there's a lot more yellow cells here compared to here. So this per se perhaps was not that novel, but we wanted to try to understand, well, what are these T cells recognizing? What are the antigens? Because by identifying this, perhaps we can replicate this phenomenon in other patients. Now, just a brief T cell 101 here. How does a T cell work? So a T cell has a unique receptor on its surface, a T cell receptor. So an individual T cell clone is marked by its unique T cell receptor. And when this receptor binds a foreign protein, an antigen, the clone expands to a population of cells. And then once the threat is cleared, it contracts and persists in the body for decades, re-expanding if the threat reoccurs. So if you in fact administer the antigen to a host and pre-populate the host with cells, you can of course protect against the re-exposure, and you are all very familiar with this concept as that underlying vaccination. But to understand whether or not such a phenomenon would be possible in PDAC, we had to first answer the question, well, does PDAC really have no neoantigens, even though it was very lowly mutated? Our thinking here was that this was likely not the case. And even if it did have these neoantigens, do T cells in fact recognize these neoantigens? And number 3, as tumors don't just have one red neoantigen, but they also have red ones and blue ones and brown ones, which of these neoantigens would be in fact the optimal antigens recognized by the immune system and thus suitable for vaccination? So to answer this question, we developed a computational model here to identify immunogenic neoantigens based upon sequencing information. And here we estimate the immunogenicity of a neoantigen based upon two parameters here we call non-selfness and selfness. And non-selfness here is measured based on neopeptide sequence similarity to other known antigenic peptide sequences derived from infectious disease epitopes. And selfness here is determined based upon how the mutated sequence differentially binds the MHC or the TCR with respect to its corresponding wild-type sequence. So in essence, this aspect of the equation here measures how similar the mutated sequence is compared to the wild-type sequence. So if it's too similar, the presumption is it would not be immunogenic. And this aspect of the equation here measures how dissimilar the sequence is, essentially by measuring how similar it is to other known immunogenic antigens. And when we apply this quality model to lowly mutated PDAC and a highly mutated melanoma and lung cancer patients, we found that neoantigen quality but not a quantity identified patients with long-term survival. So introducing neoantigen quality as a metric of tumor neoantigenicity and introducing that lowly mutated tumors like PDAC may in fact harbor neoantigens that are both clinically relevant and potentially therapeutically targetable. Now, this was bolstered by the observation that when we identified these rare long-term survivors of PDAC and when we looked in their blood for any evidence of whether the immune response persists, what we were able to find was the presence of individual T-cell clones that recognize these high-quality neoantigens. These T-cell clones were enriched in patients' primary tumors, and we can in fact identify the identical T-cell clones that recognize these high-quality neoantigens in primary tumors persisting spontaneously in the blood for over a decade. Now, so in this graph, what I'm showing you here is peripheral blood from a long-term survivor that is stimulated with high-quality neoantigens for 3 weeks. Each circle is an individual T-cell clone. And when the clone is blue, this indicates that this clone is expanded upon in vitro stimulation with the neoantigen. When the clone is colored red, that clone is also present in the patient's archival primary tumor. So you can see there's a lot of antigen-specific clones that were also present at the time the patient had a tumor with the same clones persisting spontaneously. So suggestive that natural immunity persisting for a decade later in PDAC survivors. Now, this was phenomenological because, of course, we did not have a short-term survivor group to understand what the comparison here was. So to try to understand additional evidence to whether these neoantigens were bona fide T-cell targets in PDAC, what we reasoned was, well, if these high-quality neoantigens were truly under immunological pressure with T-cells in primary tumors, then as these tumors evolved out to recurrence, then you would have selective loss of cancer clones harboring the high-quality neoantigens, but not of cancer clones harboring the other neoantigens. So in this diagram here, the cancer clones harboring the red highly immunogenic neoantigen, since these are under immune attack selectively, when these primary tumors evolved out to recurrent, you would lose cancer clones that harbor the red ones, but not the blue or the brown ones. So to answer this, we examined how 9 primary tumors in long-term survivors evolved out to 22 recurrent tumors over a decade. And we compared this to how 6 primary tumors in short-term survivors evolved out to 33 recurrent tumors for a total of 70 longitudinally matched primary and recurrent tumors over 10 years. And what we found was, although these tumors in long-term survivors had a longer time to evolve, they paradoxically evolve recurrent tumors with fewer mutations, and in fact, fewer neoantigens and fewer high-quality neoantigens. This is shown here by this score here that we call the immune fitness cost. So essentially further suggesting that these high-quality neoantigens were under spontaneous immunological pressure from T cells and perhaps were suitable antigens for cancer vaccination. So based upon these observations, our interpretation was that these long-term survivors, they mount natural immunity to high-quality neoantigens that perhaps might contribute to their exceptional outcome. But what was also interesting was that unselected PDAC patients did in fact harbor neoantigens even though they were not mounting natural immunity to these targets, thus raising this question whether immunity can be therapeutically induced in these patients when it does not occur spontaneously, as in the case of long-term survivors. Now, to induce this immunity, when we examined the nature of these neoantigens, what we found here was that these neoantigens derived from somatic passenger mutations that were individual to every single patient's tumor. Thus, for a vaccine, this would require custom vaccination for each individual patient. So back in 2017, when we were designing this, we felt the best technology for rapid custom cancer vaccination in the clinic was to use RNA. So to answer, can you phenocopy the survivor state with bespoke RNA vaccination, we approached colleagues in biopharma who were using RNA vaccines to treat other cancers such as melanoma. And we proposed that although they might think pancreatic cancer was the least likely cancer for neoantigen vaccination, because of some of our findings, this is in fact perhaps the ideal cancer for RNA vaccination. And rather than apply these drugs in the metastatic setting, which was the current practice at the time, that we should move this earlier into the adjuvant setting to phenocopy the immunological state of the host that we had observed in long-term survivors. So we essentially proposed that we should test the hypothesis that these RNA vaccines targeting neoantigens could, in the adjuvant setting, so after surgery, expand T cells to delay a PDAC recurrence. And this led to this investigator-initiated phase 1 clinical trial where we targeted to accrue 20 surgically resectable PDAC patients on whom we performed surgery to remove their primary tumors in New York. And then within 72 hours, we shipped the tumors to colleagues in Mainz, Germany, who then sequenced the tumors, identified the neoantigens, and custom manufacture individualized vaccines for each patient that include up to 20 class 1 and class 2 restricted neoantigens. These are delivered as 2 uridine RNA decatopes in lipoplex nanoparticles intravenously. Now, while this is ongoing, back in New York, we treat patients with atezolizumab, a single dose PD-L1 blocking antibody, followed by 8 consecutive priming doses of patients' custom cancer vaccines, followed by modified FOLFIRINOX standard of care chemotherapy and a vaccine booster. Now, we treated our first patient on this clinical trial in December of 2019, and then shortly thereafter found ourselves conducting this complex clinical trial in the midst of the pandemic, which was posed unique challenges for this clinical trial. Of course, there were clinical and research shutdowns, given the fact that New York was the epicenter of the pandemic in early 2020. There were supply chain disruptions, which was also important for this clinical trial, which involved real-time cross-Atlantic transfer of patient material and drug, and not to mention the slight distraction that BioNTech had now to make RNA vaccines to save the world from COVID-19. But despite these challenges posed by the pandemic, we were able to not slow the trial down, but in fact took this opportunity to in fact accelerate the trial, and we completed this trial despite the challenges posed by the pandemic a full year ahead of schedule. Okay, is rapid custom cancer vaccination feasible? The short answer is yes. You can see here, compared to our benchmark time to vaccination, which was 9 weeks after surgery, our achieved time to vaccination was within 3 days of benchmark times. So rapid custom cancer vaccination is feasible, fully integrated into a standard clinical workflow, and even after complex cancer surgery that is required for pancreatic cancer, and despite doing it in the midst of a pandemic. So if you can do it in a pandemic for pancreatic cancer, it's doable. Okay. Can these vaccines induce T cells in pancreatic cancer, a lowly mutated cancer historically thought to not contain mutated antigens suited for vaccines? So to answer this question, we use 2 tests to measure vaccine-induced neoantigen-specific T cells. A primary test, which is standard ex vivo interferon gamma ELISpot, where we take patients' blood, we re-stimulate them with vaccine antigens, and we examine whether these T cells can produce interferon gamma, or a custom assay to identify vaccine-induced T cell clones using a TCR-V beta sequencing, so essentially sequencing the T cell receptor. This is a custom assay that we developed in our lab that we call CloneTrack. So this first assay here answers, is there an immune response? And this second assay identifies exactly the specific clones that are mediating the response. And this is illustrated in this example here, where the black triangle indicates time of surgery, the blue triangle is atezolizumab, the green triangle is time of vaccine priming doses, the yellow is chemotherapy, and the final green triangle is the vaccine booster dose. And each individual green line is an individual T cell clone, so it has a unique T cell receptor. So you can see here that this clone, which is undetectable at baseline, increases about 10,000-fold with vaccination, reaching 1% of all T cells in the peripheral blood, and then decays only to then re-expand with boosting. So this is a single vaccine-induced clone. You can similarly identify all vaccine-induced T cell clones in this patient, and then you can add them up to calculate an aggregate vaccine-induced clonal fraction. And if you ask, well, are these clones really specific to vaccine neoantigens? So to answer this, we purify peripheral blood at a peak expansion, and then we re-stimulate the peripheral blood in vitro with vaccine-encoded neopeptides. We sort and sequence the in vitro activated cells, and we examine the clonal overlap between the in vitro activated and the in vivo expanded clonal pool. And then for select clones, we identify the full-length alpha and beta the alpha and beta chains of the T cell receptor. We put it in another T cell, and we examine the specificity of the T cell receptor based upon epitope mapping. So you can see here that in the prior slide, I showed you 28 clones expanding in the blood. What we find here is that of these 28 clones, 16 of these clones are confirmed neoantigen specific in vitro, and these clones recognize this particular epitope that is presented on this particular HLA allele of this patient. Okay, so essentially when we see a vaccine-induced immune response, we know that this immune response is being specifically targeted towards antigens in the vaccine. It is not a non-specific response to either the RNA or the lipid. Okay, so with these 2 criteria of ELISpot and CloneTrack, we adjudicate patients to be a vaccine responder if they have a positive ELISpot and a positive CloneTrack. And with these criteria, what we find is that these individualized RNA vaccines targeting neoantigens induced robust T cell responses in 50% of unselected PDAC patients. We also find that this vaccine-induced T cell response is primarily composed of CD8 T cells of quite substantial magnitude. You can see here reaching up to 10% of all T cells at a peak expansion and includes CD8 T cells that recognize both single as well as multiple neoantigens. Okay, so so far I've shown you for is there natural immunity to PDAC? We believe the answer to this is yes in some cases. These are the long-term survivors of PDAC. But importantly, is it inducible in other patients when it does not occur spontaneously? We believe the answer to this is also yes. But of course, for this to be a successful therapeutic in the future, we need to understand whether these T cells can last long-term and whether they can not only last but preserve their functional responses. So to answer these questions, if you look at the trajectory of an individual T cell clone, you can see here it has a very unique trajectory after vaccine priming that is distinct from its trajectory after vaccine boosting, allowing you to now fit exponential curves to the observed decay dynamics after prime and after boost. And from these exponential curves, you can essentially extract a half-life for each clone after priming and boosting, and you can translate this half-life into an estimated lifespan for each clone after prime and boost. Lifespan here defined as the time it would take for this clone to fall below this detection threshold here indicated by the dotted line. And you can now do this for every single vaccine-induced T cell clone. And what you find here is that after vaccine priming, these clones have a median estimated lifespan of 1 year that increases dramatically with a single vaccine booster to an estimated median lifespan of 7.7 years, with now 20% of these clones with an estimated lifespan beyond a decade. So not only can vaccines induce T cells in PDAC, but we believe that this T cell response appears to have quite exquisite longevity. And I would also point out that this is despite patients receiving post-vaccination chemotherapy between prime and boost. Okay, but do these cells also retain function long-term? So to answer this, what we do is we obtain the blood anywhere between 2.5 to up to 4 years post-vaccination, and we re-stimulate T cells in vitro with the antigens, and we examine whether the T cells can produce cytokines. And what we find here is that among the 8 vaccine responders, 6 of 8 of these responders have preserved polyfunctional cytokine production and lytic capacity upon antigen rechallenge in vitro to both a dominant as well as a multiple subdominant neoantigens. So essentially, not only do the vaccine-induced T cells have the ability to persist potentially long-term in patients, they also seem to have the ability to re-respond to antigen upon rechallenge. Okay, so what we find here when we now look at a median follow-up time of 36 months, which is more than double the primary endpoint, a pre-specified endpoint in our clinical trial, what we find here is that vaccine responders, these are patients who generated an immunological response to the vaccine. These patients have a median recurrence-free survival that's not reached. Compared to vaccine non-responders, these are patients who received the vaccine, but they did not generate a vaccine response, no T cell response. These patients have a median recurrence-free survival of 13.4 months, suggesting perhaps a correlation between vaccine response and delayed PDAC recurrence. And we did not find any evidence that this correlation was confounded by differences in patient tumor and treatment features. Now, okay, could it be possible perhaps that these vaccine non-responders were just less immunologically fit, as evidenced by their ability to not generate an immune response to the experimental mRNA vaccine? Namely, perhaps these non-responders just had bad immune systems that was evidenced by the ability that they did not generate an immune response to the experimental vaccine. So to answer this question, we examined whether these non-responders could generate an immune response to a concurrently administered unrelated mRNA vaccine, which in this case is SARS-CoV-2 vaccination. And what you can see here is that these non-responders are able to mount equivalent humoral as well as cellular responses to a concurrently administered unrelated mRNA vaccine, SARS-CoV-2 vaccination, so indicating that differences in host immunological fitness was not a confounder of our observed correlation between experimental vaccine response and delayed PDAC recurrence. Now, why is the immune response then only 50%? So turns out that this intravenous RNA delivery platform that we use uses lipoplex nanoparticles which have charge modifications on their surface to allow for preferential delivery of the RNA payload to secondary lymphoid organs and specifically the spleen. So in preclinical models, if you splenectomize mice, the magnitude of the vaccine-induced immune response with this particular vaccine is reduced by about 50%. Now, turns out that for pancreatic cancer, as part of standard of care surgery, we perform two different types of surgery, one which involves routine splenectomy. So it was curious to note that these non-responders were slightly more enriched in patients who were asplenic at the time of vaccination, suggesting perhaps in a more appropriately selected patient population, the immunological response rate might even be higher. Okay, so another important question is, since we are vaccinating in minimal residual disease when patients don't have any visible cancer, how do we know that these T cells that apparently have the ability to persist long-term and retain function upon antigen rechallenge, how do we know that they're doing anything at all in patients? Ideally, we would want these T cells to be able to detect and kill cancer cells, microscopic cancer cells, in minimal residual disease. Is there any evidence of that? So I'll show you one vignette here from one of our patients on this trial, patient 29. Patient 29 had the second highest magnitude vaccine-induced T cell response, which is shown in this red curve here. You can see vaccine-induced T cells reaching up to 10% of all T cells in the peripheral blood. Now, shortly after vaccination, this patient had a rise in their CA19-9, which is indicated by this blue line here. CA19-9, which is a commonly used biomarker that suggests PDAC recurrence. Now, this prompted a new MRI, which indicated a potential suspicious lesion in the liver that was suspicious for a new PDAC metastasis. Now, in fact, when we biopsied this lesion, what we found was no histologic evidence of cancer, but a dense lymphoid aggregate, so lots of immune cells, and interestingly, lots of CD8 T cells, and perhaps even more strikingly, all 15 of these patients' vaccine-induced T cell clones found in the peripheral blood. So all of the clones that were found here in this red line were found infiltrating this liver lesion, suggesting perhaps that these clones were detecting a micrometastasis that was not pathologically evident. And surely enough, when we did deeper sequencing by using digital droplet PCR, what we found here was the presence of rare p53 mutated cells, which had the identical p53 driver mutation that was found in this patient's primary tumor. So we believe this was in fact evidence of these vaccine-induced T cell clones infiltrating a micrometastasis. Now, also on follow-up imaging, we see spontaneous resolution of this lesion, so perhaps these T cells not only have the capability to persist long-term in patients, retain functionality to respond to antigen, but perhaps also in vivo can detect microscopic cancer cells and eliminate them. Okay, so to summarize what I've shown you today, in pancreatic cancer, we believe these personalized RNA vaccines can induce T cells of quite substantial estimated longevity and durable function, which we believe are desirable features for all cancer vaccines. And importantly, in data that I did not have time to share with you today, we believe these vaccine-induced T cells are in fact de novo. And this is important because it does not restrict the vaccine response to patients with pre-existing immunity, and thus significantly expands the vaccine-eligible cancer pool beyond patients with pancreatic cancer. And now at a 3-year follow-up, we show that vaccine immunity correlates with delayed PDAC recurrence. Of course, this is a small single-center phase 1 clinical trial, but we think this is, it requires, certainly warrants larger testing. We have a global randomized phase 2 clinical trial that is currently in progress. But more broadly, we think the implication of our findings is that one of the fundamental challenges for cancer vaccines as a field has really been, how do you teach the immune system to potently and durably recognize cancer? And we believe perhaps that this particular combination that we have used here, vaccinating against passenger neoantigens with RNA in a minimal residual disease, is perhaps one potential solution to this fundamental challenge for cancer vaccines. And because we are showing proof of concept now in pancreatic cancer, one of the most challenging cancers in all of oncology, and by vaccinating against passenger neoantigens, which are ubiquitous byproducts present in all human cancer, perhaps this could significantly unlock the ability to use RNA vaccines targeting neoantigens across a broad spectrum of human cancers. And there are now over 50 clinical trials now globally testing these concepts, not only in pancreatic cancer, but in a variety of different human cancers. Now, specifically to also expand upon this internally at Sloan Kettering, last year we launched the Olayan Center for Cancer Vaccines with an eye to hopefully help cement the principles and practice of these next generation vaccines and precision immuno-oncology from an academic scale as well. So partnering not only also with industry, but also be able to do this end-to-end at MSK. Okay, so with that, I'd like to highlight all of the people who did the work. All of the work that I showed you today was done by Zach Sethna, Charlotte Reich, Martina Miligeti, and Pablo Guas, all postdocs in our lab. Also, I'd like to highlight our collaborators in industry, Ira Melman, Ugur Sahin, Özlem Tureki, our clinical research team, our funding sources, my long-term collaborator, Ben Greenbaum at MSK, and of course, our patients. And I'd like to thank all of you for the opportunity to share this work. I'd be happy to take any questions.
Yeah, I mean, it seems like this could be incredibly groundbreaking for all the cancers, right? Like all the, well, immunologically cold cancers. But if it's working similarly to the patients that have natural immunity, do you, I mean, I know it's maybe you haven't had the time yet, but are we still going to see the immunoediting in these patients that have been vaccinated? Is that still going to be an issue?
Yeah, so whether or not we would, what would be mechanisms of escape or resistance to these vaccines. So I didn't have time to share results from what we have seen in our experience, but in this clinical trial that we just ran, 2 of the patients who had a vaccine response did recur. And when you examine their recurrent tumors, you can see that their recurrent tumors essentially lack cancer clones that have been targeted by the vaccine. So they do have clonal escape of non-vaccine targeted cancer clones. But we think this is specifically because one of the challenges for personalized vaccination is to make sure that you vaccinate against the entire tumor, which requires optimal selection of the antigen and pairing of the antigen to the corresponding cancer clone. And we remain sort of in the exponential phase of this in the field. We don't have all of the answers for this yet. So I think the two solutions for that is better vaccine design at the outset, and if we can't get there, we may have to resample and do adaptive vaccination over time.
I guess that's what I was going to ask. If you've got new neoantigens, that might be great. Thanks.
Yep.
Oh, sure. Thanks for that. It was really interesting. I'm curious, you mentioned that you targeted your vaccine to the spleen, and I was wondering about targeting it to other cell types like dendritic cells. Is that an interesting aspect to pursue, or do you think the spleen is optimal?
Yeah. So this particular vaccine platform that we used has both organ-specific delivery as well as some degree of cell-specific delivery. So the charge modifications on the lipoplex allow for selective delivery to spleen. It's not completely selective, I would say preferential, but within the spleen, it does deliver the RNA payload to dendritic cells. So in mice, if you deplete the dendritic cells, you lose the magnitude of the immune response. But I think the one observation from this is that we are in the first generation of vaccines. Certainly, there could be other ways to be able to induce an immune response of similar potency without being so splenic dependent. So could this be done intramuscularly with other delivery platforms, other chemical modifications, other delivery systems? So I think these are all opportunities for the field, and of course a lot of chemistry involved in this.
Right, that was my ulterior motive, yeah.
Yeah, exactly. There's a lot of chemistry involved in this, which could allow for generating immune responses of similar potency with more broad application population to target.
Thanks.
Vinod, beautiful as always. So my question is, I guess, as you're referencing second generation vaccines, when you're thinking about how you're going to design these, we know that through cancer evolution, there are many processes broken that sort of may unveil different types of RNA epitopes or RNA encoded epitopes. You think like non-coding RNAs or things in missplicing or epigenetics, et cetera. So I'm curious, how are you thinking about moving beyond things that are somatic mutations to other sorts of repertoire?
Yeah. So yeah, I think we talk about this a lot, and I think the field is probably a bit still confused maybe about this, I would say, because we don't have clear answers, because there are certainly the mutated antigens have emerged as the class of antigens where we know the most about, because we know most immunologically about them, and we importantly also know that targeting them can do something in patients. So the field is right now highly focused on mutated antigens. But there are lots of other categories of antigens, as you mentioned, from dark matter antigens, from endogenous retrovirus source antigens. You have fusion antigens. You have even also the historically used but now unfavored self-antigens and cancer testis antigens and so on. So can these also serve, have some role to play? I don't think we have the answer to that. The one thing I would say is that probably for most of these targets, you can generate some level of transient immune response in a human, because the fact that they are all intrinsically to some degree non-self. So through vaccination or by forcing it, you could probably see some degree of an immunological response at very, very best. But in order for clinical development, we really need to anchor that to some clinical outcome, meaning pick the antigen that leads to some impact, which at this point we don't really know. We have most on the mutated antigens, so this is really where the field is going. Also because, for example, in our particular case, for the survivors, when we looked at other classes, we didn't really see much impact. We saw the main dominant impact from the mutated targets.
Thank you.
Vinod, I have a question over here. Great talk. Question about the chemo after the mRNA vaccine, and whether you checked to see if after you elicited the response to the neoantigens you chose, but the chemo obviously releases all the complete set of neoantigens in the tumor, do you ever get CD8 cells to those that you didn't immunize because you've sort of made it immunologically hot?
Yeah, that's a good question, Kevon. So if you noticed on the clonal expansion curves, you essentially saw a series of clones that started at the time of vaccination and persist, and you didn't see any new clones rising sort of in the middle. Now, so at least at the clonal level, we don't see new clones emerging. However, to check to see maybe whether we would be missing something at lower levels of detection, we have examined functionally to see we see signs of epitope spreading to other neoantigens. In this case, we don't see it in pancreas cancer. Now, I think this may have to do with the indication because these are lowly mutated tumors. So perhaps if these cancers typically have only around 40 neoantigens-ish. So if you have most of them included in the vaccine, maybe the rest of them are just not immunogenic at all. So even if you're killing the cells, maybe those targets would not be sufficiently immunogenic. In a more highly mutated tumor, maybe melanoma, a renal cell, maybe where you would see something different. Perhaps there could be spreading.
Great. Thank you.
I thank you for the talk. It's amazing work, and I have been reading your papers for a long time, and I really getting engaged into your T-cell work and the immune system work, and I really wonder how this immune vaccine to the cancer going to trigger T-cell exhaustion.
Yeah, whether or not the repetitive vaccination, because we're giving in fact 9 doses of our RNA vaccine, would induce exhaustion in the vaccine-induced T cells over time. So yeah, we have not found any evidence that this occurs. We now have immunological data for up to 6 years post-vaccination, so some data that I'm not showing you here. And we don't see that the cells that we are expanding that are presumably not, well, they're not coming from prior, they're not intratumoral T cells, but these are new T cells that are primed in the periphery. They don't get exhausted with this vaccination schedule. They continue to remain functional from all of the metrics that we have tested them with. So we think this is, we're cautiously optimistic about them persisting function over time.
Yeah, this is amazing because usually when we talk about adaptive cell therapies, we always getting the negative result because of exhaustion, and I'm really glad that this is not triggering that. Yeah.
Thank you.